The European honeybee, Apis mellifera, is one of the most easily recognized insect species in the world. Originally kept solely for honey production, honeybees now provide invaluable pollination services for agricultural crops. Recently, the honeybee population has been declining due to a series of interacting factors, notably pesticides, diseases, parasites, and habitat changes associated with reduced floral quality and diversity. In temperate climates, colony losses are reported after the winter when beekeepers assess their apiaries or functional hives. In many parts of the world, overwintering losses are on the rise. In the United States, overwintering losses are also high. The varroa mite and the deforming virus have been directly linked to large-scale colony losses. This parasitic mite acts as a vector for the virus, which in turn affects individual bees and the colony as a whole. The worldwide spread of Varroa destructor, which began in the 1950s, facilitated the dissemination of virulent strains of deforming virus, causing a global epidemic in honeybee populations. To understand the current pandemic, it helps to consider the evolutionary origins of the host, the European honeybee, the vector, the varroa mite, and the pathogen, in this case, deforming virus. Apis mellifera, the European honeybee, is now found on every continent of the world. This species, however, is native to Europe, Africa, and Western Asia. Recent phylogenetic studies provide key information about the origin of the virus and the mite. Deforming virus appears to have been present in European honeybees before the introduction of the mite. However, the virus is likely found in much lower levels and with reduced virulence. It is also believed that the deforming virus was originally highly diverse, with many genetic variants. Many of these non-lethal variants of the virus have been found in the remaining varroa-free islands of Hawaii. The varroa mite co-evolved with the Asian honeybee, Apis cerana. The native range of this bee includes large and diverse areas of Western Asia. The original geographic distribution of Apis cerana and Apis mellifera did not overlap, thus the mite and the virus were not originally found together. The varroa mite that attacks the Asian honeybee is known as Varroa jacobsoni. This mite is extremely similar morphologically and genetically to the deadly Varroa destructor that affects European honeybees. During colonial times, from 1600s to 1800s, European honeybees were transported by settlers to their new homes in the Americas, Australia, New Zealand, and many other regions of the world. This initial global expansion of the honeybee and the associated deforming virus involved long journeys on cargo ships. The colonies transported during these early years, although carriers of the deforming virus, were not affected severely by this pathogen. Sometime during the mid-1900s, managed colonies of the European honeybee were transported to East Asia and for the first time came in contact with the Asian honeybee. Varroa jacobsoni, the mite attacking the Asian honeybee, had considerable genetic diversity, and two variants were able to adapt to parasitizing the European honeybee. These two haplotypes became jointly classified as a new Varroa destructor. During the second global expansion of the European honeybee, colonies infected with the mite and the virus were transported out of Europe into many parts of the world. Varroa destructor was first discovered during this new wave of colony movements. Colonies infected with a combination of Varroa destructor and the deforming virus experienced severe health problems as the mite vectors lethal strains of the disease. Research conducted in Hawaii showed a rapid change in viral prevalence, with more colonies being affected, a tremendous increase in viral levels in each individual bee, and a strong selection in favor of the virulent strains. The deforming virus epidemic appears to be a case of a part of a global trend of disease reemergence, where a previously non-lethal or low-level prevalence pathogen is suddenly problematic. In the case of deforming virus, the new vector Varroa destructor produced a large-scale viral change in the European honeybee population and triggered massive colony losses. Although definitely not the only problem affecting bees, this disease compromises the bee's immune system, making them more susceptible to the effects of pesticides, bacterial and fungal diseases. In addition to the negative effects that deforming virus causes to honeybee colonies, there is evidence that the deforming virus can affect other pollinator species. The virus can infect native social bees, such as the bumblebee, and solitary species, such as leafcutters. Deforming virus has also been found in both pests and parasites of the honeybee and in the pollen of flowers visited by them. The impact or severity of the viral spillover is still unclear and may be species-specific. Much work is still needed to fully understand the impact of Varroa from a pollinator community perspective. The remaining Varroa-free areas provide a natural laboratory in which to examine the viral diversity and viral levels without the negative impact of the mite. Protecting pollinators, including managed honeybees and native species, is a pressing goal that will require collaboration from scientists working in a variety of fields.